The slides I'm going to show today are what was originally a 1-hour lecture to general practitioners and parliamentarians that I gave a week ago in Tokyo, and that was with translation. So I think if I squeeze it down, it'll be about 30 minutes. What I'm going to do is stop in a few places and maybe elaborate on something that was relevant to the invention process. So I always try to start out my lectures with a picture of the local neighbourhood. So this is actually in the main street in Tokyo, across from the palace, and there's the Imperial Hotel. This is just up the road. This is outside a bank there. Talk about Helicobacter. So it's called Helicobacter because it's helical, like a corkscrew. So there's different. Most of the Helicobacters you see will actually look like a wine corkscrew with 4 or 5 or 6 up to 12 twists and with flagella sticking out of each end. The Helicobacter pylori's only got 1.5 wavelengths. So when you're classifying the appearance of these bacteria, you'd say, you know, the length and the diameter. So that is really the same size as an E. coli. It's about 3.5 microns. It's 0.6 microns thick. And it's got 1.5 wavelengths in it. So that's the characteristic appearance. So once you've recognised it, you can find it anywhere pretty much. And it's very easy to find it in the stomach once you start looking for it. That is a 3D image that I paid my son $400 to do with his graphics program when he was in college, which was probably more than 20 years ago now. It's still the best one on the web. He chose that it was green with like a fungusy sort of a surface texture on it, and he complains that he doesn't get any royalties. There's probably 1 million of these somewhere around. And I said, well, I did pay you $400 and I give it free off my website. But you can scroll for minutes about Helicobacter, because after we discovered it, Warren and I were interested in, you know, let's focus down on what the disease is, which is ulcers, which is 10% of the Western world. So in any adult audience, there would be 10% of those people would have experience with a peptic ulcer at some time in their life, and probably 5% of the Western world was carrying some ulcer medication in their pocket, Zantac or Tagamet, which is to lower the acid level, which could keep the ulcer problem under control. And as I'll tell you later, that was about $3, $4, $5 billion industry already when we made this discovery. But what we then found was that half of the population of the Earth, in fact more than that in the 80s, was already infected with it. And if you sampled people in Perth, about 30% had it. At Fremantle blood donors, about 45% of men aged 45 would have it, or people average blood donors. So it's very, very common, and it turns out that 100 years ago, probably the whole human race was infected with Helicobacter. There's all kinds of interesting permutations and combinations that we can now figure out and explain everything we ever knew about ulcers, which was the most common disease. Used to cost the Australian government about $300 million a year. So every time I get money from the government, I don't feel guilty because I know they banked 300. So have a look, if you want to look at Helicobacter on Google, you'll see the pictures of it. You'll see some pictures of where it lives in the stomach. So there's an ulcer, just like about the size of your thumbnail, and that would usually be in the lower end of the stomach or in the duodenum nearby. I'll show some more pictures about it. And this is an electron micrograph, if you can see the little flagella there. So this is a characteristic type of organism that lives in the mucus layer of the gut. And if you pick up, about 50% of the animals on Earth will have their own little Helicobacter. Dogs have it, cats have it, their own species. We found a dolphin on Cottesloe Beach, opened him up, he had some ulcers and had dolphin Helicobacter. So they're very, very common, and the exact origin of the human one is not really well understood. But it might have been important in the Stone Age for humans to be colonised with Helicobacter, and I might elaborate on that. Alfred Nobel, remember he started the Nobel Prizes in 1901, but it might have taken 100 years before people figured out what was wrong with him. Because he always had indigestion, headaches and spells of depression. So there were many, many people in the 19th century and early 20th century that spent their whole life with an ulcer. But you couldn't diagnose it unless you died from it. You get a post-mortem, or if you're vomiting up blood, they might have been able to open you up and sew up the ulcer. But of course people used to just die from it. So if you didn't have a bleeding ulcer, you could be like Alfred Nobel and spend your whole life complaining about the food. And it was nothing wrong with the food, it was just that you had something wrong with your stomach. And so in those days, and it's still true, that if doctors don't know what's wrong with you, they say, oh, you're overworked or under serious mental stress, because it's a great diagnosis. I'm not at fault, I'm your doctor, I know what's wrong with you, you're under mental stress. So now the onus is on you to cure yourself, not on me to cure you. So it was an incurable disease, you see. So anytime the doctor doesn't know what's wrong with you, he'll tell you that it's mental stress. And I hate to say it, but particularly if you're a woman, not because women are crazy or anything, but it's just a tradition in medicine that women go to the doctors more or something, and doctors will always find some stress. And women are actually quite receptive to this idea of stress because they get stressy, I have to admit. And so a lot of people went along with that and never ever challenged it. And so the research was always, how does the brain connect to the stomach? Where's the nerve? What hormones are involved? That was all the research. Hundreds of millions of dollars each year were going into that, rather than, focusing on what the real issue was. And after we did our original research here in WA, it didn't matter what kind of stress you were under. If you had antibiotics, you got rid of this bug, you were cured. And so people, I remember a person came in to me once and said, Dr. Marshall, everybody says I'm mad. You don't think I'm crazy, do you? And I said, well, you probably are crazy, but let's first focus on your ulcer. We'll give you some antibiotics. I'm not going to worry about fixing up the fact that you're a bit crazy, which many people are. Okay, so this is 1984 when Warren and I had just published one of our first little publications, Letters to the Lancet or something. I used to buy my trousers at Bowen's off the rack. Now I actually buy, you can get better sizes these days, luckily. And Dr. Warren, I was 30 and Dr. Warren was, so I was about 32 at that point. We'd been going. He was 42, I think. So I think he's 12 or 13 years older than me. Now this picture will allow you to actually extrapolate and figure out what you can do with Helicobacter. And this is where the inventions came. So although people would hear about stomach bacteria, really when you started looking at these things under the microscope and thinking about it, you realize there's absolutely nothing that parallels this in the human body. So here, the yellow parts are the cells lining the stomach, and they secrete mucus. So logically, you eat a steak and you digest it. So why don't you digest your own stomach every day? Because it's made out of meat. It's no different. So the difference is, so the inside of your stomach is like the outside of a fish. It waterproofs the fish. And then you've got a slimy mucus layer on the inside of your stomach. It's 0.2 millimeters thick. It's got carbohydrates in it, so they don't really get digested very much by acid. They're pretty acid tolerant. And it's got phospholipids in it. So if you stain it with heavy metals like osmium, you can see this very fine structure of like layers, like filo pastry, I don't know how you say it. but lots of little layers of phospholipid in the mucus. So that is why the acid, once it's in the stomach, it can't go back and digest the wall very easily. And if that barrier is interfered with, then you see you would get something wrong. You could get an ulcer. So that was easy to figure out. So these bacteria, 0.6 of a micron. So if you imagine that these things were like snails on the floor, this room is the mucus layer. And the acid's on the ceiling, over the roof. You know, so it's really, they're down here stuck on the ground. That is why the bacteria, they had to be resistant to acid initially to get there, but once they were in this location, they didn't actually have acid on them. So we could say, well, they don't necessarily require a low pH to grow, acid pH. Maybe they're just like a neutral pH. The other thing is, you see the massive numbers of them. So each cell in your stomach has got 300 of them stuck on it. So it's just like, I think it's like this, the frosty looking stuff on the Camembert cheese. You know, like it's a fungus or mold or something. So the whole stomach is like that if you are a person with an ulcer or just having Helicobacter and gastritis. So there are really massive numbers of bacteria there in your stomach. Now the normal situation is, if with any other organism, if you had it, it would kill you. You cannot go around usually with this number of bacteria plastered right onto your cells. Or you're going to fight it off. But the unique thing is about Helicobacter, you catch it when you're age 2 or 3, maybe you caught it off your mother, and then you're going to have it all your life. Okay, so obviously there's some mysterious process going on whereby it just stays there and doesn't go any further. And your immune system doesn't get active enough to eradicate it. So it sits there all your life, maybe fluctuates a bit, affected by diet perhaps, but that's totally unique. Now, if I take an example, so say you took a biopsy in the colon. You know, your colon's full of feces, which is just zillions of bacteria. You do not find anything like this. The bacteria, as I said, they're more like the acid on the roof again. There's the mucus layer in the colon. And down on the epithelial cells that line the colon, they're sterile. There's hardly anything down there. The mucus is flowing out of the crypts and it's got all kinds of chemical properties, etc. But you don't find the colon bacteria stuck all over the epithelial cells. So it's only H. pylori like this. So what I want you to realise is that if we discover a new disease, the only time anybody can do anything about it in Australia is when the government recognises it and gives a reimbursement. Until then, it's coming out of your GP's pocket. He says, oh, here, I'll give you $30. You can go and have this test. You say, well, everyone's screaming about $7, so you can imagine what $30 would be like. So it's a long way to go. You've got a long way to go from finding a disease or a new organism and being able to do anything about it just because of the modern system. So you might be able to get some enthusiasts who'll do it because they love science or something, but in general you cannot roll anything out in medicine. It really takes years, and that's one of the reasons. The other thing is, if you have a new disease, well then discovering a new disease, there's not much value in that. The value is figuring out the treatment for it. That would be good. And so if you've got the treatment, again, there's no value in that unless you've got a way for people to find lots of people with the disease. So if it's inside your stomach, when we started off, it cost you $1,000 to find out if you had it, because you had to have the scope down. You know, you have to have an anaesthetic and nurses and an hour's time in Royal Perth Hospital or something, and a scope. So when you start off, you can see you couldn't just roll out this new discovery that Rob and Warren and I had made, or a hypothesis. It wasn't easy to test because it was hidden in there, and it took a lot of resources to actually move forward with it. So you could see all of a sudden, if you're interested in it, then you're interested in diagnostic tests, and you're interested in ultimately treatments. So that's what motivated me. So here's a couple of ulcers. This is the duodenum, just past the pylorus. This is the pylorus, the valve at the bottom of the stomach. Peptic ulcer or gastric ulcer was there, stomach ulcer. And duodenal ulcers were a little bit more dangerous and more common. and they were just down here. That's all very complicated, so I always just say stomach ulcers. If you hear a news story, it's stomach ulcers, but it's not really very scientific as far as the gastroenterologists are concerned. There's a bit more to it than that, but they're close enough. This one's a bleeding one because it's got a blood vessel down here. See the black line? That's an artery or clotted vein or something. So that person was admitted to hospital for bleeding and vomiting up blood. And this is a gastric ulcer. This little black area here is the pylorus, so the food's going to head down through there. And on the lesser curve is this gastric ulcer, about 5 millimetres thick, and the stomach wall is 8 millimetres thick. So there's only 3 more millimetres to go, and it's gone through, and then the gastric contents leaks into the peritoneal cavity. So it's it's called an acute abdomen. The person's screaming, and the ambulance takes them off to hospital. And if you don't get antibiotics and opened up and your stomach washed out, your cavity washed out, you'll die from peritonitis because it just becomes bacterially infected with all the bacteria that you're swallowing down into your stomach. They just go through the hole, and you've got all kinds of infections there. So that would be the same as a perforated intestine or something, or in cowboy days, bullet wound in the gut would be the same kind of fatality rate. So when we looked at, so we said, well, we don't know where these bacteria come from. They are obviously living there in the stomach, and at this point we could not culture them. So we said, well, let's see who's got them, because in Western Australia maybe they come from Australian animals. At that time we weren't too sure whether other people had them in other countries, and we were being a bit careful because we realised it could be important. We wanted to keep control of the discovery. But at Royal Perth Hospital we studied 100 people coming for an endoscopy, and what we found out, so they had all sorts of things wrong with them. They didn't necessarily have ulcers. And so this is the beautiful thing about this kind of research. It's curiosity driven, and so what I mean by that is, gee, Robin, how do these bacteria live in the stomach? All the textbooks say acid in the stomach kills bacteria. It's sterile. Okay, so you don't necessarily believe everything you read in a book unless you can see the evidence and the fact that it was tested properly. Now, if you say the stomach is sterile, bacteria do not live in the stomach, the only time you can say that is if you've got a method which is 100% certain of detecting and culturing every single bacteria known and unknown. So unless you've got something that really does give you a way of detecting all bacteria, you can't say there's no bacteria. And so in medicine, anytime you read anything in medicine that's 0 or 100%, they faked it or they did a bad experiment or something, because nothing related to humans is ever black or white. There's always some fuzziness in the middle. So anyway, we had 100 patients, 58% had the bacteria. So that was a lot more common than we thought. We said, well, what diseases do these people have? And we looked at ulcers, esophagus had something wrong with it, or gastritis, bile presence in the stomach, because all these were related to different theories about ulcers. Anyway, the thing that popped out of the analysis was that there were 13 patients with a duodenal ulcer, and 100% of those patients had the bacteria. So I was like, 100%, no one will believe this. So we're searching then for somebody who didn't, some patient that had an ulcer without the Helicobacter, but in that series we could not. 13 out of 13 had it. However, if you had a stomach ulcer, 77% had it, 18 out of 22. And the stomach is a bit closer, so you can imagine if you're taking aspirin every 5 minutes because you've got a headache or migraines or something, those aspirin tablets are in your stomach, just sitting on the wall of the stomach, burning it. So it's quite easy to get ulcers in your stomach even if you don't have the bacteria. But the duodenum, a few inches further, is not going to have ulcers, not have aspirin or any drugs or anything sitting on it, but that was one of the more common areas of ulcers, but we could find evidence the bacteria there was inflamed. So in fact it was somewhat paradoxical, but then it made sense once you realised that aspirin can cause ulcers and bacteria. And then if you looked at this series, you say, well, there isn't anything else. If we're right, there isn't anything else. Most of them got bacteria, and then you could say, you then extrapolate than that, and you say, right, all duodenal ulcers have the bacteria. That means if you don't have the bacteria, it must be impossible to have a duodenal ulcer. They can't exist separately. So therefore you could extrapolate further and you say, therefore if we could get rid of the bacteria, probably you couldn't get an ulcer after that. So that's another extrapolation. Now, is there any evidence with that? Well, the whole community's continuously taking antibiotics. So there would have to be a few thousand people in Perth who'd had an ulcer and had their Helicobacter eradicated just by chance. And yet, where these people are not turning up with ulcers without bacteria, you see. So therefore, you know, with a number of extrapolations and a bit of logic, you could sit down and say, you would have to put your money on this, and it would be a dead certainty that it was true. And so then you start testing it out in different ways. So you move forward. So we published a paper with this data on in 1984. And so we asked this question, would the world accept the new cause for ulcers on the basis of data from only 13 patients? $3 billion a year going into the bank for Glaxo, Wellcome, SmithKline, those guys. On this side in Perth, these two guys published their first paper with 13 patients with a germ in the stomach. Like, get out of here, not going to happen. So it wasn't going to be accepted. And the thing that I can tell you is that if ever you do anything medical and you get 100%, start finding one that's the exception, just so you don't have 100%. It's very hard to get your paper accepted. The other thing is that, so the other thing is you get focused on statistics. We're all very scientific, so we want to get p-values, control groups, etc. And you come up, so this was a blinded prospective study, so there was no question that this was true. We hadn't faked it in any way, because we didn't actually know what the results were until 6 months later, and it was done by the health department statistician. And I didn't see the micro. We were all sending our results in separately, so it was a good study. But this is a problem. No matter what the p-value is, you've got two things. Obviously, you've got to convince people, or they want to see statistics and a good experiment, etc., and say, well, there's a p-value. But you know, if in their heart they don't believe it's true, they're not going to act on it or do anything about it. So there is a bit of publicity required, showmanship, you know, getting the idea out very carefully in the right way, in the right forums. That's necessary with a new discovery or anything new like that. People are typically skeptical, especially your colleagues, especially in your hometown. They're exceptionally skeptical. And so that this process that we go through of maybe filing a little patent and then doing some extra work and then putting a poster up just to stake claim somewhere, and at a quiet little local meeting and having some input from your trusted friends and things like that. That is very, very important, because I'm sure in this room, if someone came in and someone had a new invention and did that sort of thing in this group and chatted with a few people, there'd be a lot of people that would have experience, and they would say, you know, I know somebody in Germany who's been working on this and he's the world expert and a trusted friend, so why don't you go and work in his lab for 6 months? I mean, that sort of thing can happen. So you can very easily get catapulted out of Western Australia onto the global stage with this. But if you just try and do it all yourself, you don't initially have enough connections, and you don't really know where the expertise, and you don't really know where the person is that can look at your discovery or invention and say, that's a good one. That's really going to go somewhere because I'm one of 3 world experts. You don't have all those people. And once you start doing something new, so at this point we had been working on it since middle of 1981, and our first publication in The Lancet was 1984. So it's 3 years. And everyone says, well, it's too new. How could you be so confident about it? But in fact, we had been really hammering this night and day, 7 days a week for three years at that point. So we had all the answers. And your peers don't know that. You don't actually have any peers to review your work at that point. So this idea of, everyone was against me, but I knew I was right. We had actually tested our hypothesis many, many ways. Anyway, so here's the famous rejection letter. So when we submitted it to the Australian conference, they said, Dr. Marshall, for this, it was not accepted for presentation. We won't even give you a poster. So I didn't even get a poster. The number of abstracts we received continues to increase. For this meeting, 67 were submitted. We could only accept 56. So unfortunately, ours was like one of the worst ones. And they said, keep up the good work. Something like that. But we did find, proper people who really knew about this kind of thing in different countries. We corresponded a little bit, and I went to an international meeting. So 12 months after this, we were just so far above the local guys that it was very annoying for them, because I was just sort of a junior doctor in one of the hospitals. And I was saying, well, I've only seen 40 ulcers in my life, but I know 10 times more about ulcers than you do, and you've seen 10,000 of them. So that was the situation. So it's very difficult. So this was a quote from somewhere. Gastroenterologists and scientists found it hard to accept the teachings of the two Australians. Not only were we completely unknown, but we also seemed to be supremely confident that our hypothesis was correct. And so I was offered a microbiology job at Royal Perth Hospital, but by then I needed to get down into the stomach, so I needed a gastroenterology job. So I missed out on the gastroenterology job at that point, because they'd given them all out to the people who were doing the right thing and sort of working their way up the ladder, the promotions ladder. So there's no gastroenterology job. So I actually got a job that turned out to be better, a mixture of the both, at Fremantle Hospital, because at Fremantle they're a bit more, I don't know, lay back or something, and they'll take you a bit more on your personality and your name and a recommendation from one of their mates that you're a good guy and trustworthy, and not so much on the credentials written on the paper and the reports out of Royal Perth Hospital, which I probably had a few bad ones as well as a few good ones. Like moments of brilliance or something like that would be written on it, I think. So we then realised, searched the literature. So the next thing is you've got to read very widely and be interested in lots of areas of technology. And what I can realise looking back on it is one of the reasons we discovered all this when we did was there was new technologies coming up in the 70s and 80s. Lots of new technology, the beginning of the computerisation. So there were things around the hospital all of a sudden that had little computers in them. Now, the thing that had, I'll just, this is Giulio Bizzosero. But the thing that had happened in the beginning of the 70s was fibre optics. And so then you had fibre optic endoscopes. And everyone said, oh, isn't this wonderful? Whereas before, we had to swallow all this barium and take an X-ray, which was like a horrible black and white negative with very fuzzy resolution. And by looking at these shadows, we're supposed to diagnose what's wrong with people. Now, it's marvellous. We don't need barium meals. We've got this thing, we just slide the scope down and look in. We take a little photograph if we see an ulcer. That's much better. You can give the patient a little picture, Polaroid picture, and take it home. So it's a much better experience for everybody. But you see, they had a tool which they were not using. You could actually use that endoscope. You could take little biopsy samples all over the place. You measure the pH. You can take a photograph and measure the colour. All kinds of different things you could do with an endoscope, which nobody was doing. All they did was, oh, this endoscope replaces barium meals. So right now, I think we're in an exponential revolution with technology. So no matter what the thing is, what the technology is, the person who invents it and makes the machine or gadget has got a strategy and a use for it. So he's focused on that. But you take that thing and put it over here, do something else with it, or someone in playing around. And the best one is genomics that I've seen. Everybody's genomics, the human genome, it'll do all this good stuff. Well, what do you know? You can find out if you've got a fetal malformation, like trisomy, by just taking a blood sample and sequencing all the free DNA in your blood, and it's 99% accurate. So all those doctors who were making a living, or radiologists who are doing amniocentesis, all those nasty things, that's probably a skill that only 1 or 2 people in Australia will need to have in the future, because it'll all just be done on a blood test. So lots of things like that. And so it's a technology over there. It's used for something over here. And so keep your eyes on the new technologies, because I suppose that's an innovator, isn't it? You're going to say, oh, I'm going to use it to solve my problem rather than that one. So anyway, Giulio Bizzocchero, we found this reference, 1892. He was looking at dog stomachs. And he was famous because he figured out that red blood cells came from nucleated blood cells in the marrow, nucleated stem cells or precursors. So at that time he was involved in a controversy. Everyone said, is the nucleus necessary for a dividing cell? Everyone said, well, obviously not. You've got millions of blood cells, and the red blood cells don't have a nucleus. So anyway, he did his studies on anatomy and published his thesis, and he said that they come from nucleated precursors in the marrow. And just as an aside, well, this is my weekend hobby project. I cut up dogs, and they've got acid-tolerant spirochetes in the dog's stomach. So 100 years before us, he knew that bacteria could live in the stomach in the presence of acid. And everyone had forgotten about that. So here's his illustration. These are parietal cells, acid-secreting cells. The acid that they secrete is 1/10 as strong as your car battery. So you get car battery, dilute at 1 in 10. That's stomach acid, pH 1. Oh, pH 1.5 probably, but about that. So here's the bacteria. As I said, about 10 spirals in dog stomachs. He called them spirochetes. So he said they're acid tolerant, or they turn off acid secretion, and Helicobacter does that. So when you first catch it, it turns off your acid secretion. And so in the lifespan of the person with Helicobacter, the first few years they have it, they don't have any symptoms of acidity or anything, and they can't get an ulcer because their acid secretion is low, because the bugs have helped it turn off. This is a bit of Japanese stuff. Kitazato was one of the guys who studied their infectious diseases, or flick over there. So the question is, why, what's the greatest obstacle to knowledge? It's not ignorance, it's the illusion of knowledge. And this is Daniel Boorstin. And this is another thing, you come up with a new invention or a new technology. You say, I've discovered this new thing. Everyone says, we've got 8 other solutions for that problem. You know, we've got this and we've got that and that. And now we've got some other technology. We have to sell that. Everybody's got mega industries related to these things. Who's going to care about this other one? It's just one more. So the illusion of knowledge is that you think the problem's already solved and you don't think there's a problem anymore. But you don't realize the resources that might be getting allocated to solving this problem that's just part of society, if you like. So a lot of people were making money and had industries related to curing ulcers. And everybody thought ulcers were caused by stress, too much acid, spicy food, alcohol, and genetics. Because if you had an ulcer, you say, my mum had one and her father had it and her brother's got one. So it's obviously genetic running down in the family. So whenever you hear that something is genetic, you know, runs in the family, just realize there's 2 things that can happen. It could be genetic, or maybe the family always lived in that village and there's this little stream supplying the water supply, and up the stream is something else, and you're all drinking the same water for generations. And so it looks like it's running in your family. Everybody in, you know, that side of the tracks is a bit retarded or something. I don't know what's wrong with them. They're kind of crazy. Then you find out years later that there's lead everywhere, or chromium or something. So that's the lesson. So Helicobacter is one of these things. You catch it off your mother. She caught it off her mother, going back 100,000 years. So it travels with the tribe, and you've got your own little strain of Helicobacter, and so you may or may not get ulcers depending on other factors. So whenever you hear that something is genetic, look for the evidence. You want to know what's the mutation in what gene, and what happens when you have a knockout mouse with that gene and you put it back in, take it out, etc. Can you turn this disease off and on? So that is where we are at the moment. So a lot of these things that we think, or some of these things that we think are genetic, will fall by the wayside because we would not be able to find a gene for them, and we'll find some environmental thing that may be an infection. So the good one at the moment, of course, neuroscience and Alzheimer's disease, it's probably some virus that you get as far as I'm concerned. Maybe you got it when you were 5 and it set you up for something in the long term. So that's the illusion of knowledge. If everyone knows the answer, they're not interested in the new technology or the new discovery. So this is a little bit about my experiment. It was written up in a comic by the company that invented the antibiotic that cures Helicobacter, clarithromycin. So this is the Abbott company, the biggest antibiotic company in the world. But when they had the drug approved, they created this comic and they sent it to every doctor in America, 200,000 copies or something like that. There's only 2 copies of this comic left in existence. I've got one. Everybody probably threw it out. So here I am at Fremantle Hospital drinking a brew of Helicobacter to see if I would get infected by it and would develop an ulcer. So that's what I wanted to find out. That happened in 1984. So I felt a bit squeamish afterwards, and then a few days later I started having some vomiting attacks, and then I got better. So by 2 weeks I was over it. So the thing about Helicobacter, the reason you don't remember it when you don't remember catching it is because you caught it when you were 3 years old. You vomited for 3 days. Your mum gave you lemonade and milk arrowroot biscuits till you felt better, and then you're okay. But you had the germ there in your stomach for the rest of your life, and so you're always susceptible to ulcers. So this is the crucial inspiration. Here I am having my endoscopy. Dr. Warren's putting the scope down me, and I'm thinking, if only there were a simple, quick test to diagnose this bacteria, then I would be spared this burdensome procedure. I wouldn't have the $1,000 procedure. I have something else. So obviously there was a need there, and you could easily get a cost-effective case for it. So to make my life easy, I didn't want to be doing millions of endoscopies. I needed to be having a non-invasive way. That's my stomach with the bacteria in it. I'll skip over that. This is some epidemiology stuff. I wanted to speak a little bit about diagnosis, because this is where it gets a bit interesting, if I can find it. I'm not going to talk about Einstein. That bit about Einstein is how Nobel laureates copy everybody else's results. So this is where we started off in the early days. Is that going to work? Come on. Okay. Okay. So this thing, how can bacteria survive in the human stomach and resist acid? So the theme of this talk was how curiosity-driven research results in innovations and discoveries. And so that you don't know where you're going to go. If you know that's where you want to go, you've already made the invention, and it's just that moving the technology forward, perhaps. But if you're doing curiosity-driven research, you'll find the solution for something you didn't realise was a problem. So here we're studying how can the bacteria survive in the human stomach and resist acid. All other bacteria are killed. So I actually did this experiment, and I had dilute acid, put different bacteria in it, and after 30 minutes I cultured them out. And Helicobacter survived, and all the others were killed. Now what was going on here? So it turns out that your gastric juice has urea in it. This is urea. So this chemical here is a waste product of protein. It's how you get rid of ammonia out of your system so you don't fill up with it. It's toxic. So you package it, 2 ammonias and 1 CO2, you package it up in your liver and excrete that in your urine. So that's urea. And it's used for other things as well, but essentially a waste product. So every body fluid is it has urea in it, maybe a concentration of 2 millimoles per litre. So Helicobacter uses this. So it's in your saliva and in your gastric juice. So by having a urease enzyme, Helicobacter breaks down the urea. And on this side of the equation, he's going to soak up a hydrogen ion. So that's acid in your stomach. So nearby the Helicobacter, there's urea going in, and all of a sudden it's sucking up a hydrogen ion and keeping the pH neutral. And on this side, generating ammonia and bicarbonate. So in the test tube, you could actually show that by putting a little bit of urea in that dilute acid, you could put Helicobacter in there, and 30 minutes later they're still happy. You get them out and culture them, whereas all the other bacteria would be killed. So that explained how it survived in the stomach. So then I was looking at this and I said, well, that's very interesting. There's a lot of bacteria in the stomach. I wonder if you could just take a little biopsy and you could like put it in this kind of a little solution with some urea and some buffers or something, and it should generate ammonia, and then you get a pH change. You could just have a litmus in it, it would change colour. So anyway, the first day that I did this, I had the new technology, this diagnostic test being performed by the secretary at Fremantle Hospital endoscopy. So we would come out with the biopsy and give it to her, and I'd say, do this and this, put in this test tube, and after 20 minutes decide if it's changing colour. If it's changing colour, you'll say it has got Helicobacter. And we'll be gastroenterologists. We'll look in the stomach and take photographs and look at it very closely, and we'll try to tell whether the patient's got Helicobacter by looking. Anyway, she got 9 out of 10 right, and we got like 5 out of 10 or something. So you could not actually tell if you had the infection unless you did some kind of test. And so all of a sudden we had a rapid diagnostic test. Didn't require much skill. Anybody could do it. Is robust, foolproof. So I went home at that point, and so I went and read various books, old chemistry books and old books about the physiology of the stomach. My parents had a swimming pool, so we got some urea from Bunnings or somewhere, garden urea. It's just a fertiliser. And we got some gelatin, and we got my dad's pH testing kit with buffer tablets. We mixed up a little brew, and before you know it, we were producing a diagnostic test like this in my mum's laundry. So there was a lot of tweaking. And at that point, I met someone who was connected with Delta West, and Mel, the innovation operation here in the Curtin Incubator. So someone connected me, and I connected up with Delta West, and then signed up with Delta West, and they said we'd be interested in developing this and making it better and commercialising it. So I then signed off, and I said, can you give me some money? You know, I owe my dad $3,000 because I've patented it. They said, oh no, we never give anybody any money. It's just if it's okay down the end, we'll give you some royalties. Anyway, that was a big mistake for them, because that $3,000 turned out to be pretty insignificant compared with the royalties I got much later. But that's the usual situation. When there's risk involved, people just don't like to invest in it very much. But Delta West did improve it, and this test, it's way out of patent. But there are knockoffs all over the world. There are millions of these tests done every month, probably around the world. Maybe 20 or 30 million a year would be done around the world. You can make it in your mum's laundry using the pool chlorine kit. So it's easy to make and knock off, etc. But the original one here in Western Australia has been purchased now and is made in Utah by Kimberly Clark, the clinic's tissue people. It's in all their catalogues all over the world. And it's like Band-Aids. So this is now a lesson, a very, very important lesson for you young inventors. So I had the patent and the license to Delta West, and we did everything we could. The idea with a patent is you really got to squeeze every little ounce of value out of it, because a few years later, things that you didn't think were important will become important. But you know, at that point, my dad paid $300 and registered trademark, CLO test, and called CLO test if you like. We registered that trademark, and that test is now still sold under the CLO test, and we handed that trademark over with the patent. And the things like Band-Aids, everybody knows it, and they'll say, you know, go to a different hospital, oh, I want to do a CLO test. oh, we know what that is, you know, so they'll go and get one out of the fridge for you. So the biggest value of that is now the trademark. The patent expired 10 years ago or 15 years ago. So I just warn you, keep hold of that trademark and don't say anything about it, but keep it in your name and keep the ownership, because when your patent fails, the person you license to will say, oh, patent's expired, end of royalties, see you later. You'll say, okay, you can just go down the street. I'm just going down to China now, and we're going to make some flow tests. And they'll say, and Kimberly Clark will say, oh my God, we don't own the trademark. They'll forget about it. But so you hang on to the trademark and you'll get a reduction. It's fair to have a reduction in your royalty, but it'd be very nice if I still got, you know, 5 cents off every one of those, which could have happened. The second thing actually is as an afterthought. I found out that I was getting like 10 cents off these when they came out through Delta West. But they were selling like hotcakes in America. I found out the salesman was getting $1. So the thing was, the Delta West was selling them for $2 or $3 US. The distributor in the US was selling them to $4 for $4 to his reps, and they could sell them for whatever they could get. And so most of them were selling them for $5, and so they were getting $1. And if you had a big city like Philadelphia, you only needed as one product. You made a living off it. So that's the other thing. Try to get connected to the sales end of it as well, if some way, because that becomes a long-term value for you with the trademarks. So the next thing is, okay, that's all very well, that test, but it still needs this $1,000 test. You still got to put the endoscope down, take a sample in the stomach to put in that little test. So for gastroenterologists, it was great because you didn't have to have all these skeptical pathologists and other people around you looking at the biopsy say, we don't believe it, and they wouldn't be trying very hard, especially in America. You could do this test. And so people in America started buying it straight from Delta West, you know, FedEx packages of 50, that kind of thing. And doing it themselves in their private practice and paying for it, because what happened was you could make the diagnosis before the patient left the endoscopy room. So not only did you have the diagnosis, you didn't spend $200 getting pathology done, histology or cultures, and the patient didn't have to come back for another follow-up visit or anything. He was already walking out the door with his prescription of antibiotics. So it saved, every time you did one of these tests and went positive, you saved everybody about $250. So in America, where people paid for their health care and everyone was trying to figure out why was it so expensive, they soon figured out that there was value in this kind of thing, and a lot of health companies then said, we'll reimburse it, $30. But sadly, that doesn't happen in Australia. There's all kinds of tweaking socialistic things in the marketplace in Australia, so that you don't actually have free enterprise in the health system, and people don't know how much things cost. You invent something in Australia that's for health, and they say, well, that's free anyway. So what would be the value of that? So it's a little bit sad that you stifle innovation, and I would say there is hardly any innovation coming out of the health system from doctors, people who work in hospitals. And this is one of my little pet peeves about things at the moment. So I'll do the second part of this, is how do you do a non-invasive test? Well, hang on a minute, what about if you swallowed the chemical and we picked it up coming out of your body in your urine or something like that? So the chemical there is the urea, and we label it with a radioisotope. So this is a picture which I can draw, but no one else but Dr. Warren and I are really thinking like this, because everybody draws these big cells and they draw a little tiny skinny mucus layer, and then they draw these drawings about the cells, and the ratios are totally wrong. So we have got the cells down here. We've got a mega thick mucus layer, if you like. And so we know the pH at the top of the mucus is 2 and pH at the bottom of the mucus is probably 6, the same as these cells. We know that all the Helicobacters like to hang around down in the bottom end of it. Now what else do we know? We know that the enzyme that breaks down urea is denatured in acid. So if you've got urease enzyme from other bacteria coming in to your stomach from your mouth, that's all up here being digested all the time. So when you measure urease in the stomach, the only urease that's still working is going to be this bit down here at the bottom of the mucus layer. If it's got acid anywhere near it, it wouldn't be working. So what we did, we took starch beads, like hundreds and thousands if you like, and we coated them with isotope of urea, which is, the urea is there, and so the isotope is the carbon. So it can be carbon-13 or it can be carbon-14. And the one that I stuck with was carbon-14. It's radioactive, but the calculations are that it's such a trivial amount, it's less than 1 day of exposure to background. So people worry about it, but it's cleared for pregnant women, children, anybody. So what happens, this bead will stick in the mucus and expose these Helicobacters to that urea, forming ammonia and CO2. This CO2 will go into your blood and come out in your breath in a couple of minutes. So that is the breath test for Helicobacter. You swallow the capsule with these beads in it, and within 5 minutes you start breathing out this isotope if you've got Helicobacter. It's so black and white that it's as accurate as a biopsy for histology. So once that happened, you know, the thing just took off exponentially because GPs, gastroenterologists don't like using antibiotics. They have no experience with them. They don't really have any infectious disease knowledge. So they were a bit worried about antibiotics. Who prescribes antibiotics to every second patient that walks through the door? GPs. So their threshold for giving antibiotics is way, way down. And give them something like this, they say, great, we're just as powerful as gastroenterologists, we're going to cure all the ulcers. And once this happened, once you had these tests available, really within a few years, ulcers just kind of disappeared. And nowadays, you don't actually find many people around who have ulcers. And I can tell you, I do my research now not on humans. I have to get the Helicobacter out of the fridge, the one that I saved from 10 years ago, because we can't find any new cases, so it's rather rare. This is the breath test that's the commonest one in Australia. This is a lady takes the capsule, 10 minutes later she blows up the balloon, and we have a little distribution centre in West Perth, and that company's got, gradually getting territory in the Middle East, eventually practically everywhere in the world. And this test, we have got the trademark, so not letting that out. But again, that's out of patent as well, and there's knockoffs. But it's a funny thing, it's very difficult to have a radioactive product approved in medicine, because I mean, it's radioactivity, it's very scary. But if you do your science properly, and you can show that it is safe, it's just really almost like background, and put in several years' work, expensive work to get this sort of thing registered, it's not easy for someone to come in with a knockoff, because they still have to spend $250,000 and employ a couple of PhDs to write the documents for 12 months. You really have to invest in that before you can get a registration in Australia or any other country. And so there are knockoffs around the world, in China, etc. But those products, it's very hard for them to get into Australia because they have to find somebody who's going to stake the money to get that part. So in some respects, having a long timeline, a long, expensive, difficult timeline, which with high-level activities like PhDs, postdocs doing the work on it, does protect your product in the long term, because somebody just can't start it up in their laundry, as I did with the original test. They have to really invest in it to get to that point. So I will pause now for questions, and we can talk about ins and outs of all this.
Yeah, I have one question.
Yeah.